Hi, my name is Win, and I study neurodevelopmental science in Dr. Lorraine Lanier's lab. I think it's important for us to look at development and understand it because development is a very complex process where a lot of things can go wrong. And when things go wrong in the development of the brain, which is what I study, uh, diseases could form later on in life, such as Parkinson's, Alzheimer's, and schizophrenia. And if we could understand how these diseases form, then we could potentially prevent it in a development or find cures for it through drug therapies. Specifically in my lab, we look at the development of medium spiny neurons, which are neurons that make up 90% of the striatum. And the striatum is part of the dopaminergic pathway. And when there's not enough dopamine or the dopaminergic pathway is damaged, the Parkinson's can develop. And when we look at medium spiny neurons, we wanted to understand how their arborization pattern uh, is influenced by the environment as they developed. And when we grew medium spiny neurons in vitro, we found that their arborization is not as complex and not as dense as medium spiny neurons found in vivo. And so when we wanted to test the influence of the environment on the growth of medium spiny neurons, and when we added dopamine to these neurons, we found that the arborization of these neurons became more complex and actually match pretty closely the arborization patterns of medium spiny neurons in vivo. And so we wanted to understand how dopamine function and increase this arborization. So we looked at the canonical pathway of D1 and D2 receptors, which are the two main receptors activated by dopamine. So D1 receptors, when bound to dopamine, activates a GSG protein. So D1 and D2 receptors are actually G-coupled protein receptors. And so the GS protein, once activated, activates adenylyl cyclase, which turns ATP into cyclic AMP. And cyclic AMP is a secondary messenger that activates a whole transduction pathway, which would activate specific genes downstream of the D1 receptor. The D2 receptor actually works in the opposite direction of the D1 receptor. And when the D2 receptor is activated, it activates a GSG protein, which inhibits adenylyl cyclase. And so we wanted to see how the D1 and D2 receptors play in the development arborization. So we added agonists for D1, agonists for D2, and agonists for D1 and D2 receptors. And in all three of those experiments, we found that the arborization of these medium spiny neurons actually did not match the arborization and complexity in terms of complexity and density with medium spiny neurons found in vivo. And so we looked at alternate pathways and ideas of how dopamine functions in these medium spiny neurons. And a growing body of evidence has suggested that dopamine dimerizes D1 and D2 receptors to activate a whole new pathway called the GQ pathway. And in the GQ pathway, D1 and D2 binds to dopamine and they dimerize, and then the GQ G protein activates phospholipase C. And phospholipase C cleaves something called PIP, which turns into IP3 and diacylglyceride, and IP3 releases calcium, and through the help of IP3, calcium, and diacylglyceride, phosphokinase C will activate, which will cause another transduction pathway. So this new pathway is more complex, and the interaction of D1 and D2 receptor dopamine is less understood. So it's difficult for us to prove that D1 and D2 receptors couple. So instead, we want to prove how the GQ pathway can cause the arborization that we see with dopamine. So what we did is that we added a DREAD to the receptor to the surface of these medium spiny neurons. And DREADs are designer receptors exclusively activated by designer drugs. And so we added the DREAD to specifically activate the GQ pathway, and we added CNO, which is the exclusive drug for that receptor. And when we added CNO and the GQ pathway was activated, we saw that the arborization pattern of medium spiny neurons were actually very close to the arborization seen in vivo. However, it was not quite there. So there's a few questions left to explore here, and is the dosage correct? Or are there other pathways that dopamine might be functioning under to activate this arborization pattern? And it's important for us to look at the different methods that dopamine and other drugs influence development, because we could eventually produce a drug to rescue neurons from the underdevelopment of arborization and possibly drugs for diseases like Parkinson's.